great to be here. I'm really glad I'm going to share this time with Paul Offit as well. And, the history of vaccine technologies and the evolution of technologies is an absolutely fascinating story that's been built on increased knowledge of science, application of new technologies, some serendipity along the way, but it's always been driven by a tremendous focus on improving public health. And everybody has that in mind, and it does result, and it has resulted in tremendous advances for patients and people all around the world. So it's really great to sort of go through this. So I'm going to sort of set the stage for sort of what these technologies are that are behind the vaccines, and then Paul's going to pull through, okay, with that in mind, how actually do we develop a COVID-19 vaccine? And then we'll take time for questions as well. This is the second time I've had the opportunity to be at the vaccine boot camp. I got as much out of it last year as I think the fellows did as well, because your questions are really fantastic and made me think about a lot of things. So really looking forward to the Q&A. But in the interest of time, let's get started. We tend to think of sort of the beginning of vaccines in May 1796 with Jenner vaccinating James Phipps against smallpox. But it really wasn't until 100 years later that the real science of immunology and what became sort of the modern vaccine era really began. And Edward Jenner in 1796, he had no knowledge at all of microorganisms. And it wasn't until the end of the 1800s, the beginning of the 1900s, that microorganisms became identified as the cause of infectious diseases. There was the discovery of various cells that ingest microbes, and those are phagocytes. The very first Nobel Prize in Medicine was awarded to von Behring in 1901 for his discovery of antitoxins. And the antibody theory was also given the Nobel Prize to Paul Ehrlich in 1908 for his theory that antigens were the target of the immune response, the generation of antibodies. And all of these discoveries really led to the modern concept of natural immunity to infection. And the knowledge that we can have immunity to infection was really what drove the ability to discover and innovate and develop new vaccines. And the way that vaccine development has occurred, it's really come in waves of new technologies. I'm going to go through these on the slide, and I'm going to go through a few more, and then sort of where we are today. And then I'm going to end with just some ideas about where this can take us. And then Paul's going to show you how to actually develop vaccines. So the first technological wave we want to talk about is really from the 1930s, which is the empirical approach to vaccination, which all came about from our ability to grow viruses in cell culture. And these discoveries occurred in the 1930s and in the 1950s, the ability to grow viruses in cell culture. And once we could grow viruses in cell culture, we were able to do things with those pathogens. We could weaken them. Those are the so-called live attenuated vaccines. So attenuation is the word that means to weaken. And we could take, for example, a virus that's cultured from the parotid gland of somebody with mumps. Now that comes from human tissue. We can put that into cell culture, and we can pass it and pass it and pass it in different types of cells, going from humans to animal cell cultures. And these pathogens will then become weakened because mutations will occur that grow best in this abnormal or foreign cell culture media. And then these pathogens become weakened. They maintain their ability to produce an immune response, but they do not maintain their ability to cause disease in humans. So these are live attenuated vaccines. And examples of this that is still used today are measles, our mumps, our rubella vaccines, our rotavirus vaccines, of which Paul Offit is one of the innovators behind our reassortant live attenuated rotavirus vaccines. And this technology is still used today. Another way to take pathogens that we can grow in cell culture, viruses, and rather than weaken them, we could actually inactivate them, or we could kill them with chemicals or with heat. And then we have the whole pathogen. It will induce the immune response to the whole pathogen, but the pathogen can cause disease. And examples of inactivated virus vaccines are, for example, our inactivated polio virus vaccine, which was the basis of a Nobel Prize in Medicine in 1954, the ability to grow poliovirus in cell culture. So we're still using this technology today. And in fact, some of the COVID-19 vaccines that are in development are utilizing these technologies where they're taking the whole pathogen and attenuating it or inactivating it and using that as the basis of the vaccine. Now, another approach that can be taken is rather than using the entire pathogen that's been inactivated or weakened, what if we were to take pieces of the pathogen, a particular antigen in mind, and we can do that through recombinant DNA technology. So I remember when I was a medical student in the early 1980s, and our professors were talking to us about the incredible advances that were going to come to biomedical sciences through the discoveries of genetic engineering from the 1970s. And the first time that genetic engineering approach was really applied to vaccines was with using recombinant DNA vaccines. So the approach here is that we identify the antigen of interest, the virulence determinant, the part of the pathogen that we want to mount an immune response to that we believe will provide protection against disease. In the setting of COVID-19, we believe that virulence determinant, that antigen of interest, is this spike protein. In this case, with hepatitis B virus, it was hepatitis B surface antigen. And we had the genetic sequence of that antigen. That sequence was put into an expression system. In this case, it was yeast. Other expression systems that you use today are, for example, insect cell lines with baculovirus. But in this case, hepatitis B surface antigen was inserted into yeast. The yeast then become the factory to make the antigen. The protein is expressed and then it's isolated through the process, and that becomes the vaccine. Hepatitis B vaccines using recombinant DNA technology, that's essentially a subunit of the pathogen. This was the very first cancer vaccine. We tend to think of human papillomavirus as the cancer vaccine, but indeed hepatitis B vaccines were the very first cancer vaccines. And so that's the process that's used when we can know the knowledge of the pathogen. Now we take the antigenic sequence of interest and express it in an expression system and then produce the vaccine. We call these protein subunit vaccines. Now, another technology I want to speak about is the technology from the 1980s. Actually, I'm going to go back for a second and let you know that this technology using recombinant protein vaccines is being used for COVID-19 vaccines. There are a number of groups who are using recombinant protein technology to make the spike protein and then are formulating that usually with adjuvants in a COVID-19 vaccine. In the 1980s and 1990s was the production of a new technology using polysaccharide-conjugates. This technology takes the bacterial polysaccharide, which is the antigen, conjugated to a protein, which induces an adaptive immune response with long-term memory. And I just show that here graphically, where if we take a polysaccharide only, a sugar only vaccine is the antigen in gray, we get a very low antibody response, a B cell response that doesn't last a long time. And we get virtually no T cell response, no cellular immune response to polysaccharide only. But if we conjugate it to a protein, we get a very robust antibody response that's long lasting and a very robust cellular immune response, a T cell response that's also long lasting with memory. And this is the technology behind your Hib vaccines, your pneumococcal conjugate vaccines, and the meningococcal ACWY vaccines. And it was this technology that turned me on to wanting to become a vaccine scientist. It was in 1987 that the FDA licensed the first conjugate Hib vaccine. And I was a resident at the Children's Hospital of Philadelphia, where Paul was one of my attendings. Thank you, Paul. And every week in the hospital in 1987, we would have 1 or 2 or 3 children under age of 5, typically even under age 2, who were admitted with sepsis and meningitis, other complications of a disease called invasive Hib. And in December 1987, the FDA licensed the first conjugate Hib vaccine. And by the time I was a third-year resident in 1989, Hib disease was virtually wiped out of the hospital and wiped out of the United States. It was a miracle. And I witnessed how a technological advance created a tremendous advance for patient health. It was truly, truly remarkable. Polysaccharide conjugate technology is not being utilized for the COVID-19 vaccine development programs because the polysaccharides are sugars on bacteria. And of course, the pathogen behind COVID-19, the SARS-CoV-2, it's a virus. So this technology is not being utilized for COVID-19 vaccines. Now, what I've been speaking about with taking pathogens, weakening them, or killing or inactivating them, or selecting out subunits and growing that antigen in cell culture, that's sort of the classic approach to vaccinology. Some people call this inactivate the pathogen, first isolate the pathogen, inactivate the pathogen, and then inject the pathogen. We could call that, let's say, Vaccinology 1.0. Well, what if we went to Vaccinology 2.0, where our knowledge of human immunology instructs how we develop vaccines? And that is what's happening right now with COVID-19, where we can take the knowledge of the pathogen, know its genetic sequence, identify antigens of interest through bioinformatics, and then select the antigens of interest, and then develop a vaccine. So it's working backwards. Rather than take the pathogen and then figure out if it works, we start with knowledge of immunology, human immunology, structural vaccinology, and use that knowledge to then create a vaccine specifically and then test that it works. We sort of discover, characterize, validate, and then test. Now, as we move into the new technologies, we move into the technology with reverse vaccinology. And I just want to mention to you, because I shortened up my slide, so reverse vaccinology utilizes this approach of designing vaccines based on knowledge of genetic information and on human immunology. And the first time this was used with the FDA licensed vaccine was for meningococcal group B, where the pathogen genetic sequence was determined. There were 2,158 genes. Using bioinformatics, they were able to select open reading frames that would likely code for proteins that would stick out of the surface of the pathogen. And then they expressed 600 of those proteins that had a hit in the bioinformatics test. They expressed 350 of them in yeast, and then now they had 350 new proteins that they could test. And then working backwards, they found a few of them that produced a type of immune response called a bactericidal antibody response. And ultimately, 3 new antigens were discovered that became the 3 of the 4 antigens in one of the MenB vaccines. And this technology, reverse vaccinology, coupled with structural vaccinology, is becoming widely utilized today in development of new vaccines. So what's happening today? Today, it's an explosion of new technologies. There's unbelievable opportunity with innovation built on scientific knowledge, driven by insights and data, using big data approaches as well to develop new vaccines with the use of adjuvant systems, with the use of DNA and RNA vaccines, which are completely synthetic, which I'll touch about in just a minute, the use of vectors to deliver the antigen of interest to the patient to make an antibody response and an immune response. As I mentioned, reverse vaccinology coupled with structural vaccinology, so 3M electron microscopy views of an antigen to discover the correct epitopes to mount an immune response to, and then the use of these platforms. And this is helping us develop faster, simpler, and more efficient technologies, and you're seeing that translate today with the rapid development of safe and effective, hopefully, COVID-19 vaccines. So let's just touch in on a couple of these technologies. One I mentioned are adjuvants. So what are adjuvants? The word comes from the Latin word adjuvere, which means to help or to aid. In the late 1920s, early 1930s, there was a French veterinarian named Gaston Raymond who was inoculating horses with diphtheria and tetanus toxoids. And he noticed that when an abscess formed around the site of his toxoid injection in the horses, he got a better yield of his serum. He had a higher yield, something was helping. There was an adjuvant effect. He started to experiment where he would inject his toxoids with tapioca, with cornstarch, with mineral oil, with tree bark extracts, and he was getting a higher yield. Something was helping. In 1932, thereabouts, a British immunologist named Alexander Glennie discovered that aluminum salts had an adjuvant effect, and they were the first human licensed adjuvant was aluminum salts, and they've been widely used in billions of doses given to humans safely and effectively over the last 100 years in our vaccines. In the more recent era, in the last 2 decades or so, based on our knowledge of human immunology, we've been able to take substances that have adjuvant effects that can drive an immune response in a very specific way, targeting a Th1 response, for example, or targeting an antibody response. And what have these adjuvants been able to show? Adjuvants have been able to show that we can overcome poor immunogenicity against very complex pathogens. So HIV, TB, and malaria are immunologically complex pathogens. The world's first malaria vaccine that's right now in WHO pilot study in sub-Saharan Africa is a recombinant DNA protein vaccine with an adjuvant system. Adjuvants, in particular with human papillomavirus and with herpes zoster, using recombinant DNA subunit proteins with adjuvant systems, have been able to show that these vaccines can have a very long duration of action. And that's very important because to address one of the issues around vaccine hesitancy is if we can have vaccines that last longer, then patients will have more confidence that the vaccines are there to protect them over a long period of time. Another thing that adjuvant systems have been able to do, and this is very important for COVID-19 vaccine, is they've been able to show in the case of hepatitis B and with pandemic influenza that you can result hopefully in fewer doses in your regimen. That's been shown with hepatitis B, or in vaccines that can use less antigen per dose. That was the case with H5N1 and with H1N1 influenza. And in the case of COVID-19, where there's 7 billion people on the planet who may need 14 billion doses of vaccine, if we can get more doses of vaccine out of our yield, we'll have more doses that can get to people faster. And adjuvants have an opportunity to work on that. And also, adjuvants have been able to show safety and efficacy in important populations that are potentially immunocompromised due to therapy for their underlying medical conditions, or in people who have immune systems that are not working as well. So the very, very young who have an immature immune system, or the elderly in our populations who have age-related decline in immunity. Our immune systems decline as we age. Adjuvants have a positive impact there. I mentioned reverse vaccinology and now how it's being utilized with structural vaccinology, where we can apply structural vaccinology to antigens that are identified by reverse vaccinology. And the example here that's in most sort of advanced clinical development is the work that's happened with respiratory syncytial virus. It's been known for quite some time that the F protein on respiratory syncytial virus is the potential antigen of interest. But the initial vaccines were not effective, or perhaps weren't even safe, because the neutralizing antibodies that were produced to the F protein were being produced against the post-fusion form of the F protein. And research scientists using structural vaccinology, particularly led out of the NIH, and I believe one of your speakers was Barney Graham this week, or will be one of your speakers, his lab and his colleagues were able to show that they could keep the F protein in a pre-fusion state through various techniques. And the pre-fusion state of the F protein expresses the epitopes that will likely generate higher levels of the neutralizing antibodies of interest. So this was the situation where using knowledge of the antigen and then structural knowledge of the antigen down to the molecular level, the subatomic molecular level, could identify how to create the antigen so that it would potentially have the best effect for patient care, both safety and efficacy. As we move on with our technologies, I want to mention the concept of nucleic acid vaccines or synthetic vaccines. You've been hearing about these with the COVID-19 vaccines, with the RNA vaccine from Moderna and from Pfizer. And also there's a DNA vaccine that's made it into clinical studies now from Inovio. And the concept behind nucleic acid vaccines is that they're completely synthetic. There's no manufacturing that's taking place in cell culture outside of the human body at all. They're manufactured in a cell-free system. The sequence is essentially injected into the human body, and the human body becomes the factory that makes the antigen of interest for which the body mounts its immune response. And because there's no requirement for cell culture manufacturing, it's very scalable and flexible and fast. And in fact, it was the mRNA vaccines that went into clinical trials first. And so here's just a cartoon for how the nucleic acid vaccines can work. We can take either a DNA sequence or an RNA sequence, often delivered in a carrier system, like a lipid nanoparticle. It's injected into the human body, taken up by our cells, and then the cells take that sequence and begin to make the antigen of interest for which our body mounts an immune response. Another technique that's making it now into clinical trials with the COVID-19 vaccines, you've heard about the vector technology with the work being done by Oxford and AstraZeneca, and also now by Janssen, is we take a viral vector, which essentially becomes a delivery system for a genetic sequence. And the vectors are well recognized by the human body as foreign, and we mount an immune response to the vector. So it's essentially a self-adjuvanted vaccine that can deliver the antigen of interest to the human body. And here's a cartoon with viral vectors. They can be either replicating viral vectors that are non-pathogenic, like a measles vector, or they can be non-replicating viral vectors, like with the adenovirus vectors, either human or chimpanzee virus vectors. And then they are delivered to the human body, and then the body becomes the factory that makes the antigen of interest. And these technologies are now being utilized, as mentioned, with the Oxford AstraZeneca vaccine, with Janssen's vaccine, and Merck says that they'll be coming with a measles vector and also with a vesicular stomatitis vector vaccine for COVID-19. And then lastly, before I finish up with just concluding remarks, I want to mention something about the platforms, because I'm sure you've heard about the platforms. So the idea behind the platforms is, first, let's talk about traditional technology on the left. When we make a measles, mumps, rubella vaccine, we have to grow each of those viruses in a cell culture system in a factory that has 3 separate production lines for those 3 viruses, and then grow them up and then formulate them into a vaccine. When we make another live attenuated vaccine, for example, a rotavirus, we have to have a completely different process, completely different way of growing up the virus, or either 1 or 5 different types of rotaviruses, and it's another completely different approach. But what if we were to have a scalable approach? So for example, with the messenger RNA technology, you could just have the same technology, but switch in a different sequence. So that's what was happening. The mRNA technology was being looked at, for example, with MERS or with SARS-CoV-1. And now you could take that exact same technology already in place and just move in a different genetic sequence. This is one technology that's scalable and can be very flexible. It's sort of like an on-demand technology, like a plug-and-play technology, and can move very, very quickly. And it's built on previous knowledge to make it even more efficient every time it's run. And these platform technologies are very disruptive and are truly disrupting the way that we make vaccines, and will be making vaccines in the future, this sort of on-demand solution. So as I finish up and pass over to Paul, I just want to mention one thing just for you to think about, and we can get to this when we get to our questions. All these technologies are fantastic, but what can they do to help our society, right? Because everything that we do has to be about benefiting the patient. So traditionally, we have thought about vaccines as far as like an age approach. So vaccines for babies, vaccines for toddlers, vaccines for adolescents, vaccines for adults, vaccines for the elderly among our adults, and in recent times, vaccines for pregnancy. That's a tried and true approach, and that's not going to go away. But there's more that we can do now with these disruptive technologies. We can start to think about vaccines, as we're seeing with COVID-19, about vaccines that can respond to diseases that are associated with poverty, diseases that will tremendously impact low and middle income countries, emerging infections as we see quickly with COVID-19, vaccines that impact people who are at risk for travel or associated infections, vaccines for chronic diseases, vaccines for people who are at risk for hospital-acquired infections, be it nosocomial pneumonia or diseases associated, infections associated if they have their rib cage opened or a hip replaced, and vaccines that could be immunotherapeutic. So vaccines that could help treat and ameliorate disease by manipulating the immune system in a certain way that our body can perhaps treat itself against that disease. So vaccines that could address neurologic disorders, addiction, etc., cancer, for example. So I believe that these technologies that are incredible, the history of vaccine technology is something to be truly, truly very proud of. We can now take it to a new level and truly improve society. So at this point, I'm going to pass it over to Paul, who's going to sort of talk about how all this comes together in actually developing a vaccine and bringing it to patients. and then we'll take questions.
I'm confused about how these new nucleic acid vaccines are made. They have to be encapsulated, right, to increase bioavailability? How are they actually produced, and how is that scalable?
Yeah, so in general, the RNA vaccines are encapsulated in some delivery mechanism. What's being used most now are lipid nanoparticles. The DNA vaccines do not need that, but it seems that they, at least the one that I'm aware of, needs to have something to help it sort of get into our bodies through electrophoresis or some other way to deliver the antigen through the skin into the body. But what makes them scalable is that there's a system put in place that knows that it can deliver through like a self-amplifying system the genetic sequence to the body, and the body will recognize this, and then it will literally pick up the sequence. I'll go back later to the cartoon, and it's just a delivery mechanism that allows the body to see the sequence, and then our own molecular systems see the sequence and then start to make it. So in the case of DNA, it's in the nucleus. In the case of RNA, it's in the cytoplasm. But the system delivers the sequence so that the body can recognize it. Often it's with some help, and it will literally start to replicate it and start to make it. So once you have that, for example, with this S protein for COVID-19, you could put a different sequence into that same system. And that's what makes it scalable, because it's the same manufacturing process with the exception of the sequence. And so from a regulatory standpoint, you could show how that process is made. You don't have to repeat that every time. You're using the exact same process. It's just a different sequence.
But what is the manufacturing process if you're not using cell fermentation? How do you create them?
So I will have to get back to you with a paper on that. I can't explain it to you other than to say there's nothing grown at all. It's just delivering an instruction set to the human body. And then the human body uses its own mechanisms to actually read the instruction, in this case, the genetic sequence, and make the antigen. Paul, maybe you can explain better?
Yeah, I think large for the mRNA or DNA, it's largely synthetic. So the trick with it, and Len had it exactly right. The problem with messenger RNA, it's very labile molecule, so it needs to be delivered in a complex lipid delivery system so that it breaks down less quickly. DNA, it needs to enter the nucleus, therefore it has to be delivered by electroporation, which is a little painful. But largely these are synthetic vaccines, and Len had it right. These are plug-and-play vaccines. They're very easy to initially construct. They're very easy to scale up, and that's why they're the first ones you're seeing.
Okay, well, Dr. Offord, let's turn it over to you.
A typical vaccine timeline is about 15 to 20 years. Usually you do preclinical studies in experimental animals like mice or ferrets or monkeys or Syrian hamsters. You inoculate them with the virus you're interested in this case it's SARS-CoV-2. You hope that they develop signs and symptoms similar to human disease, and then you see whether or not your vaccine idea works. It's so-called proof of concept studies, and the advantage of this also is that you can, frankly, literally dissect out the part of the immune system that is associated with protection against disease. So it's an indicator. It's not in any sense a proof in a real sense, because mice lie and monkeys exaggerate, but at least it gives you an idea of how to head. And then you go to phase 1 trials, which are largely small dose ranging trials between 200 and 100 healthy volunteers. A lot has been made of those small dose ranging trials for Moderna and the AstraZeneca vaccine. That's what you read about in the pretty much in the newspapers and the online, et cetera. But it's really very early in the process. Then you go to phase 2. So now you think you have a dose of vaccine that works. You have a concept that works. Now you put it into hundreds of people to make sure that it is consistently immunogenic and that it at least is consistently safe, so that you can say it doesn't cause a common severe side effect. But you still don't know whether it works. The only way to know whether it works is to do a large phase 3 trial. The kinds of trials that we're talking about now involve 20 to 30,000 people who either get a vaccine or don't, and that's the only way you can know whether something is effective. Although there's much made about neutralizing antibodies associated with this vaccine, you don't really know whether that's associated with protection until you do an efficacy trial. This usually takes about 15 to 20 years. With this vaccine, however, it's much faster. for a lot of reasons. One, there's never been more money or more interest or more ideas on how to make a vaccine. There's been more than 120 investigations to do drug applications to the Food and Drug Administration. Actually, that number should probably be more than 100 companies across the globe are interested. The BARDA, the World Health Organization, the Gates Foundation have all given billions of dollars towards this effort. And that's really the key. I mean, it's when companies make a vaccine, they make progressively bigger bets. So there's sort of $1 million or so for phase 1. You go to tens of millions of dollars. Then you go to hundreds of millions of dollars when you're in phase 3. And then you go to hundreds and hundreds of millions of dollars while you mass produce the vaccine. What the government has done is they've said, we'll take the risk out of this for you. We will, at least for some of these companies, we will pay for your phase 3 trials. We will pay to mass produce this vaccine at risk, meaning not knowing whether the vaccine works and not knowing whether it's safe. No company would do that. And so by doing this, that really, more than anything else, has shortened the timeline. And just the only point I want to make here is that the one thing you can't truncate are phase 3 trials. I mean, this is, although we've done, in some cases, not preclinical trials at all, but, very quick preclinical trials, small dose ranging trials. In many ways, we sort of skip right from phase 1 to phase 3. The thing you can't skip is phase 3. I mean, this is the proof is in the pudding. Phase 3 is the pudding. Okay, so the virus, there are 4 known human viruses. They've been circulating for many, many decades. They were first identified actually in the early 1960s. I would say 15 to 20% of the viruses, the respiratory virus infections that we see come into Children's Hospital of Philadelphia every year are human coronaviruses. There are 3 novel coronaviruses, meaning no one's ever seen before, which you've heard about: SARS, MERS, and now SARS-CoV-2. This is a cartoon of the virus to simply show that there are 4 proteins. It's a bit similar to hepatitis A virus, and it is about 4 proteins. It's a relatively small virus. The key protein is the protein that sits on the surface of the virus, the so-called spike protein. This is where the virus gets its name, corona from crown. And the key part of that spike protein is the receptor binding domain. That's the part that attaches the virus to cells. And so if you can prevent the virus from binding to cells, then you can prevent the virus from infecting cells, or said another way, you can prevent the virus from infecting you. So all the efforts, everything Len talked about, mRNA, DNA, replication defective simian and human adenoviruses, are all directed towards expressing that protein, which is to say the spike protein of SARS-CoV-2. So the strategies that were used, the Len already went through. I just want to make this point. The whole killed virus approach, the live attenuated viral approach, the purified protein approach, those are well-worn technologies. I mean, we have decades and decades of experience with those technologies. The bottom 4 technologies we have much less experience with. So if you, I guess one thing that sort of concerns, I guess, all of us is you have this novel coronavirus, this bat coronavirus, that just recently made its debut in the human population. It's only been around about 8 months or so. Yet already it's had a number of both pathological and clinical presentations that no one would have ever predicted. I mean, it disproportionately affects people who live in nursing homes. More than 40% of the deaths caused by this virus are in nursing homes. And although influenza is another respiratory virus that certainly kills people who are older and in nursing homes, it accounts for flu, it's really less than 10%. It causes a vasculitis, meaning inflammation of blood vessels, because it does affect the endothelial cells that line blood vessels, causing a variety of symptoms, one of which, called COVID toes, which I'd never seen before until a couple weeks ago, is similar to frostbite. I mean, no known virus does this. It affects children in an unusual way, causing kind of a combination of something called Kawasaki's disease, which is a multi-system inflammatory disease, and then something called toxic shock syndrome, which is as bad as it sounds. And I think the other surprise, a real surprise, is this is a respiratory virus spread by small droplets. Those viruses do not thrive in the summer months. I mean, this virus has an envelope, so it tends to be much more susceptible to heat. And it's also, because it's spread by small droplets, usually when it gets humid out, that kind of small droplet spread isn't nearly as efficient. Nonetheless, this virus rages during summer months in hot and humid climates like Florida, Texas, Arizona, Southern California. So that's also a surprise. So we're meeting that. We're meeting that virus with this sort of the sort of 4 technologies that you see listed on the bottom here, with which we have very little to no commercial experience. So I just feel like, you know, you have this difficult, elusive virus that is about to be countered with technologies with which we don't have a lot of experience. And I can only imagine that over the next 2 years, we're going to learn a lot of things that we wish we knew now. But again, there are systems in place to make sure that we're always learning as we go. Okay, so regarding managing expectations, I think regarding safety, when you test this vaccine, as is being planned, and let's say 20,000 people get a vaccine, 10,000 get placebo, I think when you do that, you can say comfortably that you don't have a common or a relatively uncommon serious side effect problem. But 20,000 people isn't 20 million people, and so you may have a rare serious side effect problem. As Maurice Hillman, who I consider to be really the father of modern vaccines, having done either primary research or primary development on 9 of the 14 vaccines that we give to infants and young children, said it best, quote, "I never breathe a sigh of relief until the first 3 million doses are out there." And that's only going to be a post-licensure, or in the case of SARS-CoV-2, a post-approval or authorization phenomenon. But there are systems in place to know that. I mean, this is a great thing about vaccines. I mean, they're given to healthy children for the most part. So you want to make sure, or healthy adults, and you want to make sure that they, you know, that if once released, that you can detect whether or not they have a rare serious side effect problem. So there's the vaccine safety data link, which, you know, is a computerized linked medical record system that involves tens of millions of people, as well as something called the post-licensure rapid immunization safety monitoring program, or PRISM program of the FDA, which also can, you know, look at really tens of millions of people. So you can see who got the vaccine, who didn't, and if there's a rare serious side effect that comes up, then that will pop up in one of those systems. So we're always looking. It's really too bad, frankly, that this doesn't exist more on the drug side. It would, I think, were this true on the drug side, you probably would have known that Vioxx was a relatively rare cause of heart attacks much quicker than we did. Okay, regarding efficacy, the question comes up, what percentage of the population would need to be vaccinated, assuming that your vaccine worked? It really depends on 2 factors. The R0, which is the contagiousness factor. The R0 roughly for this disease is 2, meaning if you're infected and you're contagious and you go about your normal day, and everybody you come in contact with is susceptible, you'll infect 2 more people. And then the vaccine efficacy. So you need those 2 pieces of information. The formula is R0 minus 1 over R0 divided by the percent of vaccine efficacy. So for example, if the R0 is 2 and the vaccine efficacy is 75%, then 2 minus 1 divided by 2 is 0.5 divided by 0.75, which equals 0.67, which means you would need to vaccinate about 2/3 of the American population to stop spread. Now, it's a little tricky here because that 75% efficacy probably, I think, as this vaccine plays out, is going to be efficacy against moderate to severe disease. Because these are respiratory viruses, and with relatively short incubation periods, meaning from the time when you're infected to the time when you get symptoms. And as a general rule, those kinds of viruses don't provide what we call sterilizing immunity, meaning where you're protected not just against moderate to severe disease, which therefore keeps you out of the hospital and keeps you from dying, but also sterilizing immunity, you're protected against reinfection or mild disease associated with reinfection. That's unlikely to be true here. I think in all likelihood, this vaccine will be, I think it will be effective against moderate to severe disease. I'd be surprised if it was highly effective against asymptomatic infection or mildly symptomatic infection, in which case there could still be shedding. And I think that may be a problem in trying to get the people to understand that, if that's the way it works out, that even if they've gotten the vaccine, first of all, they may be, if we assume my 75% efficacy guess is right, that 1 out of 4 won't be protected. And 2, that even those who are protected may not be protected against shedding, in which case they would still need to wear a mask, which would be, I think, hard given that people don't even wear masks now. But in any case, if the vaccine efficacy was 100%, then 50% of the population would need to be immunized. No vaccine is 100% effective. Probably the closest is measles, where 2 doses of vaccine gives you protection, about 97% protection for the rest of your life. That's the most amazing of vaccines. And if vaccine efficacy was 50%, then 100% of the population would need to be immunized. The other thing that needs to be considered, as is shown on this slide, is that there are ethnic and racial disparities in terms of vaccine uptake. And I think that when we do these studies, in these pre-emergency use authorization studies, we need to make sure that the groups, these groups are well represented, so that you have a representative population of African Americans, representative population of elderly, especially those with comorbidities, so you can say with confidence that when you go into those groups, that you have data to support the use in those groups. I think regarding availability, this is a slide that was shown by Sarah Maboué at an advisory committee for immunization practices meeting of the CDC a few weeks ago. So what she did was she at least put forward what would be their proposed priority groups, which included healthcare personnel, essential workers, adults over 60, greater than or equal to 65, long-term care facility residents, persons with high-risk medical conditions, and others. I think right now the ACIP is working in combination with the National Academy of Medicine to try and define those high-risk groups. But at the very least, she's probably right in that we're talking about 122 million people. And think about this, as Len pointed out accurately, all these vaccines that are coming out, at least initially, are going to be 2-dose vaccines. So you're talking about 240 million people. And you have to then get that out there. I mean, and it's a 2-dose vaccine. So you have to be able to distribute the vaccine at sites far more than just a doctor's office and a pharmacy. You're going to have a lot of points of care for that kind of load of vaccines to get out there efficiently into the right people and make sure that they get a booster dose a month later. Right now, that's all being coordinated through Warp Speed, specifically the Department of Defense. A guy named Paul Ostrowski is the leader for the implementation and distribution arm of this. And they have been pretty quiet about how they're planning to do this. It would be nice, actually, for them to put out their plan now so that we can coordinate now with states and localities and districts to how to get it out there, because it's going to be a tour de force to do that. Much harder than just providing masks, PPE, contact tracing, or testing. This is going to be the hardest of all. Okay, there is concern among some of us, actually a number of us who were close to this, of what we would call, I guess, the October surprise. I mean, the thing with Operation Warp Speed is that there are now tens of millions of doses that are being produced from several vaccines. That is a large and tempting pool of vaccines into which the administration could dip its hands. I mean, I think it's extremely unlikely that we would have clear evidence from phase 3 studies of efficacy by the end of October, beginning of November. Nonetheless, President Trump has said that he expects fully that we would have a vaccine by Election Day. I don't see how that's possible unless what happens is that, the administration dips their hand into Warp Speed, pulls out a couple of vaccines and says, look, we've tested this in thousands of people. It looks to be safe. The immune responses are great. And I think that, people are dying and we need to get this vaccine out there. I think that would be a mistake. You want to make sure that we have clear efficacy data before we put this vaccine out there, because there is a fragile vaccine confidence in this country. And I think that were we to do that and then find out something unpleasant, like there was a safety issue, or or the vaccine was far less effective than one would have hoped, or was effective for a far shorter period of time than one would have hoped, I think you would shake that fragile confidence, and you only get one chance to make a first impression. And these vaccines are critical. I think between vaccines, population immunity, and hygiene, that is our way out of this. And we can't, I think, shake the confidence in vaccines. I do feel better about this. This was an article that just came out a couple days ago by Stephen Hahn, who is, as was said earlier by Chris, is the FDA commissioner, in concert with Peter Marks and Anand Shah, where what they said was, quote, given the widespread potential use of a COVID-19 vaccine, transparent discussion of FDA's vaccine and related biological products, advisory committee, which is basically the FDA's vaccine advisory committee, will be needed prior to vaccine authorization or licensure to ensure clear public understanding of the evidence supporting vaccine safety and efficacy. This made me feel much better that that's not going to happen. Now, all Dr. Hahn has to do is mean what he said.
I was hoping you could just give me a sense of whether microneedles are something that have been used on vaccines in the past, and whether it's something that could be used with a coronavirus vaccine, and does that have application for where we are right now?
Yeah, it's absolutely being studied. There's some good work being done out of the University of Pittsburgh and other places looking at the use of microneedles with investigational COVID-19 vaccines. They've been used with other vaccines in the past. There was an influenza vaccine that was on the market that was actually taken off the market because there wasn't a lot of consumer interest that used a microneedle intradermal approach. So absolutely, but it's not for tomorrow.
Yeah, the only thing I would add is that the advantage of that is that, first of all, it's not a shot, which people generally don't like, especially children. It's like a Band-Aid essentially that has these 100 or so tiny little needles, and the advantage is so each needle has a little bit of antigen at the end of it, and by putting it sort of on the skin and under the skin, you basically have this antigen taken up by professional antigen presenting cells called dendritic cells, which are very efficient antigen presenters. So there's no reason this can't be a viable technology. Just a matter of doing it.
Paul, I wonder if I can just ask you a question on that, because I think people would be interested. So there was an influenza vaccine that used a microneedle approach. It wasn't a patch, but it was in the delivery system. And my understanding was that it was a consumer flop, and it was stopped being marketed because people think they need to get a shot. And so there was no consumer interest, and so it was a commercial failure, which I found fascinating, but I wonder if you have any thoughts on that, because people felt they needed a shot.
Yeah, that's interesting. I think we just probably have to do a better job of marketing.
I'm Taryn Mento. I'm a reporter out here at the San Diego Public Media Station. And Paul, this can go to you first, because you were the one that mentioned it, that made me think of this question. And I'll preface this by saying, I know that the government gives out a lot of grants for research, but I'll admit that I don't know much about the process after a technology or product comes from said research. You mentioned that we wouldn't really be where we are in terms of the vaccine development without the heavy investment of the government. So I'm curious how that all works with determining distribution, setting price, et cetera, after we achieve some sort of product. Because we've been talking a lot about who will be in control of those decisions. So I'm wondering, with the investment, does anything come along with that in terms of decision making?
Right. So as it stands, these vaccines will be provided for free by the federal government. That the administration costs would have to be picked up, I think, by insurance companies, but they're to be provided by free. And then the question is what these companies will charge the government. The initial numbers that were floated out, the only two I've seen is Pfizer and Moderna, because they seem to be leading the league right now. But the Pfizer price was about a third of Moderna's price. This is all, I mean, I'm not a vaccine economist. I'm probably not the one to answer this question. I'm really just a scientist. But it would be upsetting to me if there was any sort of price gouging associated with this, because I mean, the government really took the risk out of it for these companies. And there is some unseemliness associated with this. I mean, Glenn may want to comment on this too, because he works for a company. But I think, even though these were planned sales, when the head of Operation Warp Speed, basically cashes in stock from having been a consultant for Moderna and for whatever, 30 million. And then, the chief medical officer of Moderna cashed in stock for 40 million. It looks terrible. I mean, it feeds right into the notion of the anti-vaccine people. And a lot of the language actually that surrounds this race for a vaccine is disconcerting. I don't know who came up with the term warp speed, but it couldn't possibly have been a scientist. So between that and, sort of the vaccine race for a vaccine, who are the finalists? I mean, it gives one the sense that the corners are being cut, the safety timelines are being ignored. And so I do worry about this. I think right now there isn't a vaccine. There's a theoretical vaccine. And so I think once we have a vaccine or vaccines in hand, I think by no later, I'm sure, in the middle of next year, and we know the characteristics of those vaccines, we'll be able to explain them better. But as long as there are phase 3 trials, when we talk about a 30,000 person trial, I think Glenn alluded to this, the HPV vaccine was a 30,000 person trial. I think a conjugate pneumococcal vaccine was around a 35,000 person trial. So that's about right for a phase 3 trial. I mean, the rotavirus trials were much bigger because there had been a previous safety issue with a vaccine in the 90s called RotaShield. So those were 70,000, 60,000 person trials, but those are bigger than usual. So as long as we do the phase 3 trial, as long as we let it go to completion, as long as we have clear, robust efficacy data, then we've done this the right way. So, but I do, the other thing that worries me is that we have economic and technological advantages in this country that other countries don't have, although we don't act like it currently. I think that, we should be able to provide vaccines to other countries. I mean, how much more information do we need to know that what happens in other countries can affect this country? So I'm sure there's going to be some ugliness associated with this, but I hope I'm wrong.
Yeah, I'll add to that. And I'm going to tackle in on the global piece here. So the absolute critical importance is that vaccines are for everybody. We need to address the disparities that we see here in the United States and around the globe. This disease is going to disproportionately also affect low and middle income countries. And these vaccines have to be available for everybody. I work at GSK. GSK has stated that we're not going to make any profit on our COVID-19 vaccines, and any profit that may be made will go right back into developing vaccines for emerging diseases, including COVID-19, because this is going to go on for a while. So this absolutely has to be done right, because I think the future of public health is on the line here. We have to get this right. And it's so, so important that this is done properly. The other thing I want to say along the side of this is that for the company that I work at GSK, like the other big companies, Merck and Pfizer, we have another business in vaccines in addition to what we're doing for COVID. So at GSK, just as an example, 2 million people a day receive a GSK vaccine. That's 700 million doses distributed a year. So we have to keep that business going, because what we're seeing, and Sean alluded to this when he made his introductory remarks, we're seeing that people have not been going to the pharmacist or their doctor's office during the shutdown. Tremendous declines in immunization, especially in adults as well. Pediatric immunization is starting to come back, but adult immunization is still down tremendously from where it was. And so we're at risk here in the United States and around the world for resurgences of measles, for example, polio, and other diseases that we can absolutely control. Pneumococcal disease, there's been a tremendous rollout in making sure that conjugate pneumococcal vaccines were reaching low and middle income countries. And so there's tremendous, tremendous concern from the WHO, from UNICEF and other groups that we make sure that it's not only about COVID, but we have to make sure that the whole vaccine public health initiative gets back on track, because we've had tremendous declines. So we need to make sure that we keep the rest of our businesses going and not just only focus in on COVID. And that's why I entered my part of the presentation saying we need to take these technologies and help improve our world, because that's what these technologies can do.
Okay, so we'll take another question, but first I will say in deference to Dr. Offit's thoughts, I'm going to change the title of this session, and it's no longer called the Race for a Coronavirus Vaccine. We'll just call it the update on coronavirus vaccine. Yeah, but let's go to a question from Umair, please.
Hi, Umair Irfan at Vox. For Dr. Offit, given that we're likely to have more than one vaccine come to fruition, and that we're not likely to have a one-size-fits-all vaccine, what does that do to the distribution effort? How does that complicate it? And I mean, for instance, like if you have elderly people and you have a vaccine that's not ideally suited for them, would they have to wait until one comes to fruition, or do we distribute what we have as best as we can?
I think we need to make sure that there is an adequate representation of the various subsets who we think are going to be frontline responders. And it may be that that one vaccine is better than another for one or another subset. I mean, that may be true, but I don't think we can reasonably authorize the use of these vaccines if there isn't an adequate representation of those subsets. And let's suppose you're right. And let's suppose that the one mRNA vaccine is better than, say, a replication defective human adenovirus for an elderly population, then that would be the vaccine that they would get, which further makes it more complicated for distributing it. I think the distribution effort is going to be incredibly complicated as it stands. But I think once we're talking about sort of subsets with different vaccines, it gets even more complicated because you have a 2-dose vaccine. You have to make sure these are not going to be interchangeable vaccines.
Yeah, and we're also going to be introducing the vaccines in a setting where we have social distancing. So that's going to further complicate the implementation and distribution. So, but these are challenges that people are really starting to very, very actively think about. But I think, as Paul was saying, we have to make sure that the whole public health community comes together and solves this with the goal of making it happen, rather than, what we're starting to see out there, right? Because everybody needs to have access. That's the key.
Yeah, here's one thing that actually I do not understand. I'm not understanding the math on one thing. You've had a number of people, including companies, say that they thought they could have something that was FDA approvable by, say, the end of October, or these are 2-dose vaccines and 30,000 person trials as it stands. Therefore, you need to recruit 30,000 people who are going to get vaccine or placebo. They're not all getting vaccine on day one. I think if you were lucky, you could have everybody get 1 dose of vaccine, let's say by the end of August, which would be amazing as it was. Now you have to wait a month to give them that 2nd dose of vaccine. Now you're in September. Then technically your biggest immune response, the boost of the 2nd dose, really is about 2 weeks after that. So already you're in the middle of October. Then you have to make sure that you're protecting against disease. I mean, you're not just trying to protect against infection. The clinical endpoints for a number of these trials is moderate to severe disease, right? You want to make sure you're keeping people out of the hospital and out of the morgue. Well, that's only 20% of infections. And you, as Len said, you're really encouraging them still to wear masks and social distancing. You're not sending them all to that biker conference in Sturgis, South Dakota. You just tell them good luck. You still want them to sort of, in many ways you don't want them to get sick, but on the other hand, the only way you're going to know whether or not your vaccine works is if a critical number of people in your placebo group get sick. So when people say the end of October, I do not understand that. I'm missing something because the math doesn't add up to me.
And just to add to that, and Paul had the great fortune of being involved in one of the very large rotavirus studies. I've been involved in the licensure now of more than 16 different vaccines in a lot of different studies. The road to developing a vaccine and showing that it's safe and effective is a long road. And there's a lot of twists and turns along the way. You have to make sure also that your data is clean, that there was no breakdown in the randomization, that people couldn't figure out what they were getting, that the data are not manipulated in any way, not to say that they would be, but you have to be able to verify that the systems that collect the data are closed, that nobody could access the systems. It's highly, highly regulated, highly, highly complex for a very good reason, because at the very end, we want to know that that vaccine is safe and effective, and that every dose from every lot is exactly the same. So the chemistry and manufacturing is critical. My understanding of the first vaccine that went into clinical trials, the mRNA vaccine, that vaccine is kept at -70 to -80 degrees Fahrenheit. So the cold chain is an issue as well. And that vaccine will have to be kept on dry ice in the doctor's office, in the pharmacy, if I'm not mistaken. So there are lots of things that it takes time, because at the end, the goal, and no question about it, it has to be the goal, that what's given to a patient is safe and effective. That's the key.
Yeah, if you want to scare yourself, call somebody who was previously the head of a manufacturing division, like B.J. Samant, who was head of Merck Manufacturing Division. Ask him what it takes to make a vaccine. I mean, what he'll tell you is just to validate the processes involved in manufacture. Each aspect, how you clean the vats or tanks or whatever, if you're done, that all has to be validated. I can't see how that alone doesn't take a year. But this is Warp Speed is in many ways a black box. I mean, it's sort of co-directed by the administration in concert with the Department of Defense, and it has sort of that Department of Defense ethos, which is that they just don't tell you much. I mean, I'm on the NIH's active group. I'm also on the FDA Vaccine Advisory Committee, which Len knows about well. And they've presented to us, and also they presented to the ACIP, and you just don't get much information from them. So I just wish this was a little more transparent of a process from the standpoint of Warp Speed, but they have this kind of security issue, fear, that either cyber attack or physical attack. They talk about that a lot, and they keep things quiet.
Okay, let's get some more questions. And for the speakers, you're having great questions here, but if we could limit the answers maybe to one round as opposed to several. So we have about 10 questions we want to get to, and let's go to Corey right now.
Hi, I'm Corey Piker from Oprah Magazine, and this is a pretty straightforward question. Dr. Offit, you had mentioned that the vaccine will very likely, whichever vaccine it is, will most likely need a booster or a 2nd dose. How does the timing of a month, where does that come from? And explaining to people why they need that 2nd part of the dose or the booster, what happens if they don't get it?
Well, then it won't be as effective. I mean, when say Moderna did their trial, they gave 1 dose, found it, and then compared the neutralizing antibody titer to what people's neutralizing antibody titers were that were convalescent from a natural infection, and then found they gave a 2nd dose that they had a better neutralizing antibody response, and assumed that that second dose neutralizing antibody response would be more likely to be protective. So thus was born the notion of a 2-dose vaccine, and they gave it a month apart, which is fairly standard. You're just going to have to explain that to people. I think if they got 1 dose, there probably would be some level of efficacy, and we may learn that from the trials. It's possible if there's dropout, but you just have to be able to explain it to people. It makes it harder.
And I would say that the second dose, typically when there's a second or even sometimes a third dose, where you get this boost in the immune response, both cellular and humoral, it's the response that often is associated with durability. So there could be protection with a first dose, but it's likely to be more so and longer lasting with the second dose. And I think the durability is going to be very, very important with our COVID-19 vaccines. And we may not know that information up front because typically we would wait and follow patients for a year and report out what happened over the first year. But that's not feasible. That's not practical in this situation. So this information is going to come over time. But I think we'll obviously we're going to need to do as a community a tremendous amount of work with the US population and the global population around the COVID vaccines. I've seen data that shows that 20% of people say there's no chance they would even get a COVID-19 vaccine, and 30% say they're on the fence. And so there's tremendous amount of work that needs to be done. And a lot of it, and that's why I said earlier, like the future of public health is on the line here. We have to get this right, or we're going to take 5 steps backward. And we can't let that happen.
So my first question is for Leonard. I'm wondering if, well, last week there was a great Kaiser Health News article about how vaccines might not be as effective in people who are obese. Mm-hmm. And I was wondering if that's being considered and if there are any particular technologies, including adjuvant technologies, that could possibly address that concern.
Yes, Sony, that's great that you bring that up. So obesity triggers a chronic inflammatory state in humans and in all animals for that matter. And this chronic inflammatory state can be a negative. If our immune systems are always sort of revved up, they may not respond properly in the way that we would like in normal situation. And so obesity, especially morbid obesity, has been associated with adverse outcomes in general with people with chronic heart disease and diabetes, etc. But also we've seen with vaccines, a classic is with hepatitis B vaccines, that vaccine responses are lower in people who have high rates of morbidity, morbid obesity, the BMI is greater than 30. And we know with COVID-19 that that's exactly what we see clinically, that people who have morbid obesity have much worse outcomes from COVID-19. And in the United States, I think, I don't know the exact numbers, but on the order of something like 50% of our population is considered obese with BMIs above whatever the number is, 23 or something like that. So it's a real issue. And I'm hoping that in the clinical trials that are being conducted, that there's a reasonable number of people who have morbid obesity. However, those people tend to have comorbidities. They tend to have underlying hypertension and diabetes, and often people with comorbidities are not enrolled in the clinical trials. And so Paul's been involved with the active work. You may know more about the inclusion and exclusion criteria, but we truly hope that these patients are part of the studies up front, and they'll be reviewed by the advisory committee because it's a real question how effective these vaccines will be in that patient population. Because you're absolutely right, Sonny, those patients have a high rate of disease and they don't respond as well to vaccines.
Hi, thank you, and apologies for my video not working just now. So forgive me, but I have to ask, given what you know about all these different approaches that you so eloquently described, would you put your money on any one type or another as working? And then I have a quick unrelated follow-up question.
I'd like to firmly come down on beats me. I mean, I am no longer making guesses about this virus anymore. I mean, I don't know. I'm open-minded to any approach. I mean, we've seen I know that the Chinese now have an inactivated virus a vaccine that I know they've submitted those data to JAMA. So we'll see what those data look like. But you know, we're all in phase 1. You know, we really don't know. It's trying to read the tea leaves. I believe neutralizing antibodies are important. How long they last, how high of titers you need. I'm open-minded to any of these approaches or many of those approaches working. I don't know. I'm not putting my money on anything. Although I just like to say one thing. I have talked to Sunny Saltzman 100 times on the phone, and now I actually get to see what she looks like. There you are.
And I'm going to say that I hope they all work. We need multiple winners here. We need vaccines for 7 billion people on this planet. No one manufacturer is going to be able to scale up and make enough doses for 7 billion people. And so I hope they all work. And then over time we figure out, you know, which work better in certain situations if that's the case. But let's just hope that there's a lot of winners here.
Perfect, thank you. And then a really easy follow-up question. When there are side effects, what causes them? Like what's happening in the body?
Well, the things that I can tell you people are worried about, and the NIH Active Group has an immune pathology subcommittee. There were 2 vaccines that were made in the 1960s. One against respiratory syncytial virus, which is like this virus, that it's a viral cause of pneumonia, and then the other measles virus. In both cases, those vaccines were made by taking the virus and then inactivating it with a chemical. The chemical was formaldehyde. In both of those cases, when the children that got that vaccine, when they then were exposed to the natural virus, did worse than people who'd never gotten the vaccine, the RSV vaccine especially. And the reason was something actually that Len alluded to in his talk. Both of those viruses have a fusion protein. That's how the virus is attached to and enter cells. They actually fuse to the cell, then enter. It doesn't enter by the sort of more classical way of so-called endocytosis. So, and when they were inactivated, it altered that fusion protein, so it perturbed the immune response. The reason that's relevant is SARS-CoV-2 is also, that spike protein is a fusion protein. So the inactivated vaccine that's being made in China is made not using with formaldehyde, but with a different chemical called beta-propiolactone. But there's an understanding that you don't want to perturb that protein. You want that protein to look just like it looks on the surface of the virus. That's what you want it to look like. And when you use mRNA or DNA or replication-defective simian or human adenoviruses, it's not exactly the virus. I mean, probably the closest would be a live attenuated viral vaccine approach where you could be assured that that was true. So that's one thing that people are worried about. And then people want to make sure that what happens to this vaccine doesn't happen with dengue, which is, you know, that again, there was something called antibody-dependent enhancement, where you make antibodies to the virus, but it doesn't neutralize the virus. And those antibodies actually help bring or facilitate the virus entry into cells. People are looking at that. There are going to be systems put in place by the CDC and the FDA post-licensure or post-approval to make sure that that's not happening. But those are the things people worry about.
Yeah, and just to add to that, Lisa, just to start with, so until, of course, we get to post-marketing and, you know, millions of people are vaccinated, I think it's going to be really important with the first 30,000 people in the study and then the next 30,000 in the next study, that there's data at least 1 month at a minimum after the second dose where we can compare what we call unsolicited adverse events. So things that happen to people, we can compare them across the 2 groups and we can see if there's any imbalances. So if there's more of X, whatever it is it equal between the 2 groups? Because it probably should be. Now, things happen randomly, but with large numbers like 30,000 people, we can get a general sense if there's some imbalances, but we need to follow people and at least a month after each dose, where we basically ask people in the trial, ring us if anything's going on. Because if it's only data for a week after the dose, we're just not going to be able to have even the beginning of a sense. And what typically we do, what the FDA typically requires, is that if with a new vaccine, an investigational vaccine that's never been licensed in that class, that people are followed for at least 6 months, if not for a year. But that's just not feasible or practical given the public health crisis right now. But I would say at least a month would be important.
Okay.
Thank you.
So it's 4:20, a little after 4:20. I want to try to get through. We have 6 or 7 more hands up. So if you could limit your questions to just one of the 2 speakers, well, hopefully we can get to everybody. But we'll go to Kira right now.
Hi, thank you. Kira from Leaps Mag. I have a number of questions, but I'll try to just focus on one. At what point would you feel comfortable urging the public to get the vaccine based on the data that we will have? Is it after phase 3? Is it after 3 million doses, or somewhere in between?
After phase 3. I mean, I think if you've tested this in, let's say, a 30,000 person trial, and at least, let's say 20,000 people got the vaccine safely, you know, that there may be, you know, fever and headache and chills and muscle ache, but that, you know, that you can tolerate that. And it was effective. I mean, certainly, you'd like to think 60, 70% effective, at least for the length of time that you took it. That's reasonable to launch. I mean, I would get a vaccine if that was the case, and if my group was represented, you know, old people was represented in that subset. So I think that you don't have to wait for 3 million doses. It's not practical, really.
Yeah, but to add to that, in addition, there won't be enough doses for everybody in the United States when the first vaccine is approved. So there will need to be some prioritization, and Paul talked about this earlier, where who would, you know, when you have limited number of doses, who should get the first doses. And so there's a lot of work happening around that to make sure that this is done in an equitable way that's going to benefit the most people, which would be people at most at risk to start with. So but that's a hot potato right now as well, because everybody wants to think that, you know, they're in that group, right? So that's going to be very, very important, and it needs to be done well and explained well. While something may not be available for you, why it's important that other people get protected. And, you know, that's a hot potato, right now, to help others.
If I could just ask a really quick follow-up to that, can you comment on whether you think the ring vaccination approach should get more attention versus just vaccinating high-risk groups?
I think, you know, we're so bad at contact tracing. I'm not sure that ring vaccination would work. I mean, smallpox is a little easier, which was where ring vaccination brought that virus to its knees. But, you know, smallpox is different from this virus in that there's no such thing as asymptomatic smallpox. And so you can tell who's infected. In fact, you just look at their face and you can tell who's infected. Here you can have, you know, 80% of people have, you know, mild or asymptomatic disease and can still shed. So it's much harder. I mean, this falls more into the category of polio than smallpox.
Melissa Healy from the LA Times. You have talked a little bit about this, and maybe this is for Dr. Offit first, but the traditional vaccine design, basically based on killed or attenuated virus, seems to be what China has focused on most, and seems to be coming along seemingly fastest. Can you make the argument, aside from the idea that we ought to use this pandemic as an opportunity to try out new technologies for designing vaccines, shouldn't we somewhere in Operation Warp Speed's portfolio have a simple attenuated virus vaccine or a killed virus vaccine? I mean, okay. Could you make the argument against that or for it, or?
The problem with making a live attenuated viral vaccine is probably the most difficult vaccine to make in terms of time. You have to figure out to make sure that it's not too attenuated and that it's attenuated enough. And so it's hard. That's really a trial and error vaccine. It's not surprising that that's the last one. I know Cogenics, at least the Farmingdale, New York company, working with Serum Institute of India, was working on that. I heard. China also has a replication-defective replication-defective human adenovirus 5, as well as the inactivated vaccine. We'll see how these play out. I really don't, I wasn't being glib before. I really don't know which vaccine strategies will work the best. I'm open-minded to any of them. We'll see.
But the strongest argument seemingly then for some of these newer technologies would be one of basically manufacture and just speed.
It's speed. They're the first up because they're the easiest to construct and the fastest to make and the fastest to scale up. That's why you're seeing them first. They're, as Len said, plug and play vaccine. Just plug in the gene.
Hi, my name is Hannah. I'm with Crosscut in Seattle. So recognizing that there are a lot of things to control for when you're investigating the efficacy of vaccines across racial demographics, there are certain aspects of racism that make it difficult to say whether something is purely ineffective down to genetics. There are people from certain racial demographics that are suffering greater death and hospitalization than other demographics, and people are anxious to ensure representative inclusion of different demographics in vaccine trials. Are there examples of vaccines that have had poor demographic diversity racially and proceeded to exhibit poor efficacy in those underrepresented groups?
Certainly. The FDA really pushes to make sure that when vaccines are tested in these big phase 3 trials, that those trials represent the American population about whom to whom they're about to be given. Are there examples of, I can't think offhand of vaccines that say work well in one, say, racial or ethnic minority and not another? I can't think of that, but I could be wrong. Len, do you know of any specific example of that?
I don't know of any examples, but I will say that the vaccine development approach has not really gone down that path as deeply as it has occurred on the pharmaceutical side, where with pharmaceuticals, we can dose based on race and ethnicity, on BMI, on gender. We haven't gotten there yet with vaccinology. I believe that's coming. But what we do know is that there are certain groups who are at higher risk. And so what we of course know with coronavirus 19 that underrepresented minority populations in the United States are at greater risk with morbidity and mortality. And so we absolutely need to make sure the vaccine is safe and effective in those populations. And as importantly, make sure that the vaccine is available for those populations. Otherwise, we're not serving our patients. And that's so it's absolutely critical. And when it looks at prioritization, that's certainly a hot topic right now, but to make sure that people can access who truly are at higher risk. And so we have to make it happen. And I think over time, we'll see more and more focus on this and understanding the genetics, the immune response, and the approach of epigenetics as well. Really critical.